we've seen throughout the day yesterday and this morning the importance of maximizing the utility of every precious data point, and that's going to be even more important the rare the population is. Even within type 1 diabetes, there are aspects of the disease journey where finding those patients is not as easy. But the important thing is, what do you do with those data? How do you transform those data into something that that is meaningful? So I want to start with Justin P, who's giving me the looks. So that you saw the type 1 diabetes journey from your vantage points at the agency. And so I wonder if you can comment on that experience from a regulator and seeing the journey from data to solutions.
Okay, that one sounds a little bit more palatable for me, Klaus. So we were involved with the, Justin, Earp, and I were involved with the type 1 diabetes prevention work that was discussed earlier, and we were involved from the agency side on the assessment of it. I think it was a pretty, it was nice collaborating with you. I think there were a few lessons learned. I really like close collaboration, but I also like the idea of these formal pathways, right? So we have FFP, fit for purpose pathway. There's the critical path innovation meeting pathway. And those pathways, I like those pathways, and I think you guys might like those pathways because they represent the agency's thinking, not Justin's thinking. Justin, the modeling Justin's thinking. So I really encourage everyone to utilize those pathways. I think now that we've got two submissions from the critical path on diabetes prevention and the treatment of new onset diabetes, those formal pathways have been very smooth and nothing but a pleasure with me. And I think having formal agency pathways, we've got a system to engage not only the clinical review division, but we involve statisticians and pharmacometricians and pharmacologists. I think that has been a very helpful methodology and pathway. I don't know if that answered your question.
It does because the formal pathways provide that forum to have that interaction with not just a review division at the agency, but a whole collection of groups from the agency and get a coalesced vision on drug development tools as such, not without the review of the products being affected or intrude upon as you have those conversations. Justin, you see the entire landscape of the Fit for Purpose initiative, not just for type 1 diabetes. And I wonder if you can comment on your experience in seeing those interactions happen.
Yeah, hi. I've been with the agency since 2008 in pharmacometrics, and one of the general observations I've seen modeling and simulation have an impact across the full spectrum of drug development, right down to approval based on exposure response, which doesn't happen every day. It's been a long time since I've seen that. But your points early on about how do we get the most information for rare diseases is right at the crux of where we see clinical impact. A generalization I could make based on my experience might be that clinical impact could be a function of a combination of unmet medical need combined with the practicality or feasibility of collecting data. And, diabetes itself, the general type 2, when we think about that, has been such a frustrating area for me as a modeler to work and try and have impact because the mechanism, you can measure the endpoints and the biomarkers, and you can apply very mechanistic models. And the beauty of these models, a lot of them came forward in the late 90s, is that they're able to connect so many of these different factors together as a function of time. So because it's a function of time, we can ask what's going to happen next. And that's why we can do so much with them. But for type 1, here's your rare disease. Here is where we need to get the most out of the data points. And here's where we need to know what's going to happen next with disease progression. So this is why we have to be mechanistic. This is why we have to apply it over time and pull all of the information together.
Right, excellent. And Larissa, I'll go to you next because you see the world from the very specific aspect of the neonates, right?
Yes.
And so, can you comment on that?
Yeah. I think, when I think about the neonate, like oftentimes my mind goes to the very sick, premature neonate. And so in thinking about some of the ways to model, I think there's some difficulties and challenges because there's such both heterogeneity and the primary diseases of prematurity that we think about, like BPD and necrotizing enterocolitis, the big two that we're always trying to make an impact on. But I think my hope and view for the future is that with the expansion of real world data and initiatives to try to use some of those tools to really hopefully understand and tease out, to help us better understand the pathophysiologic underpinnings so that we can start to make a better impact on both preventing and treating these diseases.
Yeah, and we have heard that we hope to complete next year for pulmonary dysplasia is going to be at the center of that.
Mm-hmm.
So that's great. Now, I'm going to complete this round with Larissa because you certainly see the world from the biologics angle. And so, can you comment on this entire notion of extracting maximum value from data through collaborations?
This entire notion of extracting multiple. Sure. Well, of course, use of data that have been accumulated previously through various platforms in drug development is very important. We see this current trend towards the use of real world data and real world evidence observations that have been made in hospitals, hopefully in NICUs in the future, in neonatal populations, pediatric populations, taking these data and applying them to drug development, I think is going to be invaluable because of many complexities, which I'm sure we'll talk about during this panel, many complexities of doing drug development and running clinical studies in such a vulnerable population as neonates, let alone children who are still in utero. And so I think we will see more data that have been accumulated in the past to be applied to product development. And we'll also see more drug development tools that have been shown to be valid markers or outcomes in the past experience to be applied to future product development. So you had a global question to give you answer.
No, but that's perfect because that shows that even though there's centers at the agency, I mean, there's the view at fundamentally is the same about the need to make sure that you connect what is clinically meaningful to the data, to what analytics and what solutions are going to be needed for very specific applications. Joe.
So, Esther, we're widening the lens a little bit. I think JDRF has had a tremendous amount of experience over the years thinking about this prevention space, development of therapies, development of or supporting the development of modeling tools, issues around screening. So I wonder if you can comment on JDRF's experience specifically around the idea of disease prevention. And we've heard obviously some success here recently, but also many of the challenges that are still faced. So I'd like you to comment there.
Yeah, so I think I'm gonna, as we've seen, there's been a lot of progress on the staging of type 1. I think we all understand the benefits of screening and prevention of diabetes ketoacidosis at that diagnosis. We now have a therapy that can delay the onset. But I think we've learned a lot from this research screening settings on how do we move to a global screening
So we need to identify biomarkers, better biomarkers of progression. Like we know the progression now based on the antibodies on the glycemic readouts at 2 to 5 years, maybe too long. And we know that decline in C-peptide is much significant as we get to the transition from stage 2 to stage 3. So can we identify better biomarkers of progression? Can we identify better predictive biomarkers from the new onset trials? We know that a lot of this predictive biomarkers come from running clinical trials and looking at responders versus non-responders. So, maybe some inducing tolerance and using some peptides to induce tolerance in the new onset, we'll learn that maybe responders are associated to specific HLA types. So that happens after the running a clinical trial. So can we do that on this prediction biomarkers? I think we have the data. Also, I think what we learned from screening is the community engagement. As we expand globally, we need to maybe improve tools so it's easier to screen. Can we identify better ways of doing testing at home or doing sampling at home, even glycemic measures? We know in research settings that follow-up for screening, it's quite intense. And as we expand, people may come from one screen that maybe not appropriate screening. So I think we identify what is the best time for screening. I think the most important thing I want to mention at the end is for health providers, clinical guidelines. It's like as important as screening is, monitoring it is as well. And as we expand the screening programs and we educate on the benefits, I think it's very important that clinicians understand what it means if you prescribe a test and it comes back positive. How do you monitor? What do you deal with that? I think this is very well understood in the research settings. But as we move beyond that, I think that's one of the main efforts that JDRF is doing now, building this clinical monitoring guidelines on how to follow people that are at risk.
Yeah, I think that's a really important point. I mean, we've focused a lot on the interaction between developers, organizations like JDRF and FDA, but I think we don't want to forget, obviously, the physicians have a critical role. And I think, Stephanie, that raises a question for you from the patient perspective. Hearing these conversations around prevention, I'm curious if you can comment a little bit sort of on the patient perspective on benefit, on risk in the prevention setting, intervening in individuals that while the disease process is there, they may not be feeling or experiencing symptoms. So how do patients think about that in your experience?
Thanks so much, Joe. And I just want to invite everyone in the audience for a moment to look up from your screens and lean in and hear first my appreciation for the opportunity to represent the patient perspective on this important panel. I do think every opportunity we have to bring real life experience of what it means to actually live with these diseases or to care for someone living with these diseases is critically important. And I happen to have the privilege of being the mom of a young woman who's now 24 and has lived with type 1 diabetes 16 years starting next month. And I can tell you, my brother-in-law was diagnosed with type 1 5 years before my daughter was diagnosed with type 1. And we did not go through any screening at the point at which he was diagnosed. Had we gone through any screening, we likely would have identified that she also had the genetic marker that my brother-in-law has. At that point, there was no TZIELD, so we wouldn't have been able to do anything to stave off the onset. But there is so much energy and attention paid to treating post-diagnosis with insulin. And I'm here to tell you that despite what often is perceived as the panacea or a life-sustaining medication, that insulin is a really hard drug to have to live on day in and day out 24/7. And anyone who tells you that the standard of care is acceptable because we have insulin doesn't know what it's actually like to live with synthetic insulin and try to titrate that every single day. my daughter, in spite of all the amazing progress that's been spearheaded by many people in this room and certainly by JDRF, my daughter goes to bed with a lot of the same risks every night that she did when she was first diagnosed. She doesn't wake up with low blood sugars, and that means she might not wake up. So the idea of being able to stave off onset by 2 or 3 years, the idea of knowing that she has genetic markers and we can modify or moderate really would have been a game changer in our lives. And I can't stress enough the importance of assessing risk and what it means to engage the patient community in evaluating risk. And the idea, by the way, that people living with type 1 are not living with a great deal of risk when yet they walk around with lethal doses of insulin that they have to self-administer belies the reality of what it actually means to live with type 1. So when we start to think about the opportunity for intervening with TZIELD or curative cell therapies in the not too distant future, I encourage you all to really get to the heart of what it means to be living with the risks this patient population lives with every day, and dream of a time when we actually are treating type 1 as an autoimmune disease with the opportunity to intervene along the way, not treat it purely as a metabolic disease with insulin after the fact of diagnosis. And I thank you for the opportunity to share that perspective.
No, that was fantastic. Thank you. And I want to make sure that I also amplify the fact that in the type 1 diabetes space, screening is a tangible thing and was a tangible thing even before the advent of the transforming therapies that have been approved. So it can be done. Now it needs to be done more. I agree with you, but
to that point, certainly after my daughter was diagnosed, our entire family enrolled in the TrialNet study. My brother-in-law's entire family did the same. We've done the self-home test for my youngest son, who was a little too young to be following year after year at that point. I could not agree more. I think the opportunity for universal screening, because family history of type 1, we didn't screen. We didn't think, well, that same risk is going to, befall our family, even though we knew my brother-in-law had it. So if everyone was automatically screened, think of what that could mean.
Yeah, thanks very much, Stephanie. So, Jim, you've had a great talk. You've heard some of the conversation here. I'm wondering, you've seen a lot of movement from the lab to the clinic. And how do you see the connectivity of what you do in the lab to what's done in the clinic in terms of some of these quantitative tools? In other words, how can they enable that? What might you do more? And just give us your perspective on some of that. Just be sure and use your.
The rare diseases, especially those that are rapidly progressive, lethal in the newborn infant development, does create some challenges with respect to clinical development. For example, I no parent is going to want to sign their kid up to a placebo trial. Some of the interventions are such that they're quite invasive, and the follow-up is quite invasive. So we've been at Penn. I run the Orphan Disease Center, and we have a sort of broad remit, and really encouraging communities to come together around putting together robust natural history data, so that we can credibly come before an agency and recommend an open label study associated with bias for sure. But we're really early where we are in genetic medicines, mostly are diseases that are lethal in the pediatric age groups and very rapidly progressive, which is one of the reasons we're doing it. But also that gives us an opportunity for disease modification with their transformative effects. So there are some products that are not just very good that would not be amenable to that. Those products shouldn't be in the clinic right now, at least in the diseases that we work with. But just one last comment about that, which I sort of referred to before, but there was an era ERT ushered in the notion you can make money on rare diseases. And there was an era over the last 10 years, 5 years, where these new platforms were attractive for investment because they were platforms. You could generate value by literally enrolling a subject in a trial. Well, that ship has sailed. And what we've encountered over the last year and a half is an incredible exodus or retreat from financial support through investors in biotech or the pharmaceutical industry, largely in the rare diseases. So this is, I don't know if there's a precedent for this in the history of medicine where we've actually there's 50 years of recombinant DNA history. There was a celebration at Cold Spring Harbor. This is why 50 years of investment, we have solutions and we can't figure out a business model. But, so I'll conclude. What that has done is the rare disease communities are pretty mad, pretty upset. And what we're now seeing is they're trying to take control of their own destiny, getting involved in drug development. And I think this may actually create an opportunity for developing consortium with health, and health authorities are leaning into this. And you heard Peter talking the talk and walking the walk. And this may be a time where we can make progress at bringing coalitions together where we probably couldn't before. So, and that relates to working internationally to develop databases so that we know the natural history of the disease. Because we don't do that. You could have a therapy and you're all dressed up and know where to go.
Perfect. Yeah, no, this is perfect. So I think with that, we're going to conclude the panel. Thank everybody. How long?